Hello gamers, welcome back. No green screen, because I still need to fix it. Mostly the camera. The camera needs to be fixed. By that I mean replaced. I have a camera option that I'm going to replace it with, but I'll work on that later. For now, I got a couple minutes, so I may as well do some studying.
Oscillation.
Oh boy.
And glycosylation, well, you can look at the word, the prefix.
I already don't know where we are.
That is, but not all of them. So let's go through some examples. So the first post-translational modification I want to talk about is glycosylation.
Okay.
And glycosylation, well, you can look at the word, the prefix glyco tells us that it has something to do with a carbohydrate. And so glycosylation is the adding of a carbohydrate to a protein. And most of the proteins in this video are all in green. Right. And glycosylation usually happens to proteins that end up being embedded in the cell membrane. So you can see here we have a cell membrane and this protein embedded in it, and then there are these carbohydrate groups attached. So here's a carbohydrate group attached and here's another one. And glycosylation helps to identify different types of cells. And one very common example of where we use glycosylation is in the ABO blood groups. So let's take 4 different red blood cells, and let's just say that each one of these red blood cells belongs to a different person. And so red blood cells have these proteins embedded in their surface. And these proteins are going to have many times different carbohydrate groups attached to them. So let's say that this person right here has this particular carbohydrate group attached to it. That makes him blood type A. Let's say that this person has a different type of carbohydrate attached to the protein. Let's say it looks something like that. That makes them blood type B. Let's say that this person has actually both of those carbohydrates attached to his red blood cells. That would make him blood type AB. And let's say that this last person does not have any carbohydrates of this category attached to the proteins on his red blood cells, and so that makes him blood type O. So here's a very common example of how glycosylation is used in the identification of different types of cells. Let's go on to a different type of post-translational modification that's pretty similar, and that is lipidation. And lipidation is when we add a lipid to a protein, also a protein that's going to be attached to the cell membrane. And this lipid we're looking at is actually an example of a GPI anchor. And GPI anchors are lipids that help to attach or tether proteins to the cell membrane. And just to give you an idea of maybe why this would be necessary, so to quickly review the structure of the cell membrane, we have these hydrophilic heads. That means that they are polar. And then we have inside these hydrophobic tails. And that means that they are nonpolar. And so the protein has both polar and nonpolar parts on it, and maybe it just doesn't attach well to the hydrophilic portion of the cell membrane. And so this GPI anchor, a lipid, kind of plunges into the lipid or hydrophobic part of the cell membrane, and we know that substances that are similar, like substances that are both hydrophobic, attach very well to each other. So this lipid, which is hydrophobic, attaches very well to the inner part of the cell membrane that is also hydrophobic. And so that's how it helps to attach the protein to the cell membrane. And both glycosylation and lipidation usually do occur in the endoplasmic reticulum or in the Golgi apparatus. Let's move on to some protein modifications where we use glycosylation is in the ABO blood groups. So let's take 4 different red blood cells, and let's just say that each one, the prefix glyco tells us that it has something to do with a carbohydrate. And so glycosylation is the adding of a carbohydrate to a protein. And most of the proteins in this video are that look kind of more squarish, and those are receptor sites for the. Let's see how phosphorus and potassium pump. That's when you remove a phosphate. So that's how it helps to attach the protein to the cell membrane.
Okay, there we go.
And both glycosylation and lipidation usually do occur in the endoplasmic reticulum or in the Golgi apparatus. Let's move on to some protein modifications that have more to do with the activity or the function of the enzyme and less with the structure. So one very, very common protein modification I want to discuss is phosphorylation.
Classic.
And phosphorylation is basically the adding of a phosphate group to a protein or to an enzyme. And then phosphorylation comes along with dephosphorylation is when you remove a phosphate group from the enzyme with a protein. I'm just going to make a little bit of room over here. And so what you're looking at is this schematic diagram of the sodium-potassium pump that's found in basically every animal cell. So the sodium-potassium pump. And another name for the sodium-potassium pump, which is the enzyme that you're looking at, is the NAKATPase. And so this enzyme or protein, the sodium-potassium pump, is responsible for maintaining the proper osmolarity of sodium ions and potassium ions in and out of the cell. And so let's see how phosphorylation regulates this protein. So, and again, the proteins that you're looking at, they really all represent one protein. It's just we're going through the motions of the changes that happen to this one protein. But it's not like we have it's not like we're looking at 6 different proteins in the membrane. So here's our first step. And when you look at this enzyme, you can see that there are 3 receptor sites for a particular ion. This is the sodium ion, represented by these dark blue circles. And then there are 2 receptor sites that look kind of more squarish, and those are receptor sites for the potassium ions, and those are represented by these light blue squares. So those are the potassium ions. And so back to the first step in what's happening here. The sodium ions will attach to the receptor sites on the enzyme. And just to clarify, this is the intracellular space. So this is basically the cytoplasm. And so these sodium ions are coming from the cytoplasm, and then out here is the extracellular space, the outside of the cell. Anyway, back to our first step. So the sodium ions attach to the receptor site, and when the receptor sites are full, it's going to cause something to happen. It's going to cause an ATP molecule, adenosine triphosphate, to break down into ADP, adenosine diphosphate plus phosphate.
They just
and then this
switch.
phosphate will attach itself to
They're
the
like
protein.
voice.
And that is phosphorylation.
I'm so confused.
And so when this protein gets phosphorylated, when the phosphate group is attached to it, it causes there to be some sort of change in the conformation of the protein.
Like
and that change in conformation
they suddenly
causes
have
the
an
protein to turn
accent
itself
out of
around by
nowhere.
180 degrees and face the
What is
outside of
going
the cell.
on?
And that's what you're looking at right over here. So our protein is still phosphorylated.
All
I know
good,
it's in a
though.
different place, but just keep in mind that there's still a phosphate group attached to that protein. And then the protein releases these sodium ions into the extracellular space. So the next step is number 4, and again, we are still phosphorylated, still a phosphate group attached to our protein. And so the next step, the potassium ions on the outside of the cells will attach themselves to the protein on the receptor sites. And take note, there are 3 receptor sites for sodium, but only 2 for potassium. When the potassium receptor sites are full, it's going to cause a different change to happen. This phosphate group is going to be removed.
Phosphate goes in.
And that is called dephosphorylation.
Potassium goes in.
So that is dephosphorylation. And so that phosphate group ends up in the inside of the cell and gets recycled in some other way. And when this protein is dephosphorylated, when that protein is removed, it's going to cause a different change in the conformation of the protein. And that change in conformation causes the protein now to turn around again by 180 degrees, and it faces the inside of the cell. So that's our 5th step. You can see the protein is facing the inside of the cell. And the last step, the potassium ions are released into the inside part of the cell, and then we're back to our 1st step. And so you can see that the phosphorylation and dephosphorylation basically regulates the activity of this protein. And the end result that we're trying to get to is that we want there to be on the outside of the cell a rather low concentration of sodium, a rather high concentration of sodium, and a rather low concentration of potassium. And on the inside of the cell, we want there to be a rather low concentration of sodium and a relatively high concentration of potassium. And that's accomplished by the fact that for every 3 sodiums that are pumped out, 2 potassiums are pumped in.
Neurons
and so
are
here's an example,
bananas
a
in
very
the
common
sea.
example of how phosphorylation regulates a protein. And this doesn't happen just in the sodium potassium pump. It happens in many, many enzymes and proteins in our bodies and in cells.
Developed
Let's move
by
on
the
to
sodium.
2 other protein modifications that also have to do with regulating an enzyme or something similar to that.
Bananas.
So the next protein modification I want to talk about is methylation. And in particular, the methylation of certain proteins called histones.
Oh my lord, wait.
Those are these green circles that you're looking at. And histones are these proteins around which DNA wraps itself. So they're found in the chromosomes, and they help to package DNA in a very tight and organized manner.
I guess
and
we'll
sometimes
just
histones are
keep
methylated.
building
Let's
this
put some methylated
ladder.
histones. And methylating and
that takes
demethylating
us.
histones helps to turn certain genes on and off. And so here's another example of a protein modification helps to regulate activity, but in this case, we're regulating the activity of genes as opposed to proteins. And another protein modification I want to bring up is proteolysis. And by looking at this word, proteo means protein, and lysis means to break something down or to cut something. And proteolysis is sometimes to take a protein and activate it, we need to cut it. And in fact, the insulin has to be cut twice before it's activated. So let's cut this protein twice. And you may have actually heard of the term zymogen. And a zymogen is an inactive form of an enzyme. And sometimes the way to activate a zymogen is by cutting it, and that's proteolysis. There is one more protein modification I want to discuss. And that is sometimes we add a protein ubiquitin, to another protein. So that protein I just added is ubiquitin. And this process is called ubiquitination. And what ubiquitination does is it basically marks this green protein for degradation or for breakdown. So within a short while of being ubiquitinated, this protein is going to be destroyed and the different parts are going to be recycled. Let's just quickly review the post-translational modifications that we discussed. So we talked about glycosylation and lipidation. Those are 2 protein modifications that generally happen to proteins that end up being embedded in the cell membrane. So glycosylation was the adding of
I don't even know how to spell
and
that.
what ubiquitination does is it basically
Okay. Alright.
marks this green protein for degradation or for breakdown. So within a short while of being ubiquitinated, this protein is going to be destroyed and the different parts are going to be recycled. Let's just quickly review the post-translational modifications that we discussed. So we talked about glycosylation and lipidation. Those are 2 protein modifications that generally happen to proteins that end up being embedded in the cell membrane. So glycosylation was the adding of a carbohydrate group, which helps to identify certain cells, and lipidation is when you add a lipid to a protein that generally helps to anchor a protein to the cell membrane. Then we talked about phosphorylation, methylation, and proteolysis. And these all have to do with activating or deactivating an enzyme or genes. So phosphorylation was the, we brought the example of the sodium-potassium pump, which is basically regulated with phosphorylation and dephosphorylation. Then we spoke about methylation, which can basically helps to turn on or turn off certain genes. And proteolysis is the way in which many enzymes are activated. And the last post-translational modification we talked about was ubiquitination. And ubiquitination marks a protein for degradation, and then the various parts are recycled.
Okay. I have no commentary right now.
So hopefully by now you're familiar with the central dogma of molecular biology that tells us that DNA makes RNA in a process known as transcription,
Every single time we have these streams,
in a process known as translation.
to be hype
Let's
as
take a
the
look
first
at
stream.
cells. Let's say that right over here we have an eye
Bro.
cell, and let's say that we have a
Content
skin cell.
is still pretty cool.
And so
but
what makes the eye cell an eye cell,
goddamn.
and what makes the skin cell a skin cell? So they both have the exact same DNA.
There's a
All the
lot
cells in our
going
body,
on.
any organism have the same DNA. And so what makes an eye cell and a skin cell is which genes are expressed in that cell. So in the eye cell, we have the expression of genes that make certain proteins that are unique to the eye cell. And in the skin cell, we have genes that are expressed, and they make proteins that are unique to a skin cell. And so the question I want to focus on is, how does that happen? How do we regulate the expression of genes so that only those proteins that are necessary for the cell get expressed or are made? So let's just frame our question. How is gene expression regulated? So let's look at the options that we have. Maybe gene expression is regulated at the protein level. What that means is that the DNA in each cell is all transcribed into RNA, and then all the RNA gets translated into proteins. So according to this model, you have in each cell all of the proteins coded for by the entire human genome, if we're talking about humans. But then only those proteins that are necessary for the cell get activated. So for example, in the eye cell, all the DNA gets transcribed into RNA, and they all get transcribed into proteins, but only the specific eye proteins are activated, and all the other proteins are inactivated. Maybe that's what happens.
That seems
Maybe
a little
gene
silly.
expression is regulated at the level of translation. That would mean that all the DNA in the cell is transcribed into RNA, but not all the RNA gets translated into protein. Just the RNA that make proteins of that particular cell would get translated. And the third option we have is maybe gene expression is regulated at the level of transcription. And that would mean that not all the DNA gets transcribed into RNA. only the DNA that codes for proteins for that specific cell would get transcribed into RNA. So for example, in the eye cell, you'd only have DNA transcribed if that DNA is a gene that codes for a particular eye protein. And the answer to our question is that usually gene expression is regulated at the level of transcription.
Gee,
and
I
if
wonder.
we think about it, this should make sense because this is really the most effective way for a cell to make use of its resources. So let's take a look again at our, if gene expression was regulated at the protein level.
Because you'd have to
Again,
make
so we're talking
hella
about humans,
proteins
so
and
that would mean that
RNA.
each of our cells, all the DNA gets transcribed into RNA, and then all that gets made into protein.
You don't need
so
to.
we would have a tremendous amount of protein in our cells, and actually thinking about it, I
That
don't even
said,
think
our
there's in one cell
cells
all the
aren't
proteins
always
coded for the genome. But even if
super
there was,
efficient
that would
in
be
the
a huge
first
waste of
place,
energy.
so
It takes a lot of ATP to put together
who
proteins. So
knows?
why would we want each cell to make a whole bunch of proteins that they'll never even use?
Until
So this is not very efficient.
someone
What about
with
translation?
authority
If
tells
we
you.
regulate gene expression through translation, well, it's more efficient than the protein level, but it's still also not that efficient because that means we have a bunch of RNA that would never even get translated. So making RNA, it doesn't take as much energy as making protein, but still that would be a big waste of energy. And so it turns out that regulating gene expression at the transcription level is the most efficient because we're not making any RNA or any protein that we're not going to use. And we actually have a lot more to learn about how gene expression is regulated, but there's a particular model that we understand pretty well, thanks to the work of two French scientists by the name of, one of them was François Jacob, and the other one was Jacques Menard. And they discovered the mechanism of the lac operon. So we call it the Jacob-Menard lac
Yeah,
operon.
lac
And lac stands for the
operon,
lac
alright.
lactose. And the lac
Alright,
operon is found in the
we're
bacteria
seeing
E. coli.
some
So it's a
gamer
prokaryotic
terms
cell.
right here.
And the picture that
Okay.
you're looking at is the stand of the lac operon. It's a section of DNA in E. coli. And let's just label the diagram so that we orient ourselves. So let's say that this is the coding strand,
Application.
which means that this is the non-coding or the template strand. And if you recall, it's actually the non-coding or template strand that gets transcribed, and that's the reason that I color-coded the non-coding strand with various genes. Each of these colors represent a gene, and we'll explain in a minute what they are. If I wanted to be more exact, maybe I really should have also color-coded the top because these two strands are complementary to each other. But I'm not going to fill it out, just use your imagination and remember these are complementary. And I also just want to point out that I drew this transcription bubble because it's going to be easier for me to show you what's going on in that way, but the default is that these two strands are really stuck together, and you do not have this bubble forming unless transcription is happening. So just keep that in mind as we go on. Okay, so what is this lac operon? So before we talk about the details, the lac operon has a couple of genes that will make enzymes that help E. coli break down lactose. So let's take a look. So over here we have these three genes. They're called structural genes. It's not important for you to remember that. But these three genes, this here is the lac-Z gene, this is the lac-Y gene, and this is the lac-A gene. And so if you recall, the sugar lactose gets broken down into glucose and galactose. So glucose and galactose are monosaccharides, and lactose is a disaccharide. And the lac-Z, lac-Y, and lac-A genes are all each going to code for an enzyme that helps in the breakdown of lactose, or in the metabolism of lactose. So let's look at the lac-Z gene. The lac-Z gene codes for a protein, beta-galactosidase. And beta-galactosidase is the enzyme that actually breaks lactose down into glucose and galactose. The lac-Y gene codes for the enzyme lactose permease. And lactose permease helps the cell bring lactose into the cell. And the lac-A gene also codes for an enzyme that helps in lactose metabolism. We just won't focus on it because it's not as important as the lac-Z and lac-Y gene. So these genes are all needed for the metabolism of lactose. And let's just label this part over here right before these three genes. That's the start site. So if RNA polymerase was transcribing, that start site tells it here's where you should begin to transcribe. And then after the lac-A gene, we have a stop region. So that tells RNA polymerase stop transcribing. And normally, E. coli uses glucose as its energy source. That's the default. However, if glucose is not available, or if it's suddenly inundated with lactose, it will want to break down lactose. But why should E. coli express lac-Z, lac-Y, and lac-A in the absence of lactose, right? We just explained before that would be a huge waste of energy. So the default situation is that these genes are not expressed. Let's see how that is. So over here we have a promoter site. And the promoter site is the place that the RNA polymerase kind of just sits. So let's label our RNA polymerase.
Okay.
That's the enzyme that puts together RNA. So it just sits there. And after the promoter site, we have the operator site. And on the operator site, there's
This
a
is
protein
actually connected
just sits there.
to transcription,
And this is called
which
a
means
repressor.
that we need way more space.
And you can see the repressor is kind of blocking the RNA polymerase. So normally the RNA polymerase would want to proceed in this direction and transcribe this entire area of the DNA, but the repressor doesn't allow it to do that. It just sits there and blocks transcription. And so again, this is the default situation that's in the cell. It uses glucose as an energy
Man,
source.
I just
and
want to
transcription of these
include
genes is blocked.
retroviruses in my diagram. Can I include retroviruses in my diagram? Okay, maybe I can't. Maybe I need to move some stuff. Let's shift this over at least a little bit. Okay. All the pre-meds who aren't weebs will be so confused by the music in the background. Hey, welcome. Oh, even the weebs might be confused too. From BNA, brand new animal, triggers, one of Trigger shows. Pretty good, pretty fun. Okay. Now that I have more space for transcription related stuff, we can now start talking about operons, which are fun. Coding strand, terminator, intron, extrons. No, that wouldn't be as part of that, so I think we can just have that separate. That's fine. On. We have the actual Jacob monad. LAC operon, lactose operon, the famous one that everyone uses for their examples in AP, the biology, that one, you know. Right. Promoter operator start. Compressor. I guess we can talk about the different elements of the operon system. Well, not system, just kind of a line of stuff happening. We got the promoter. Comes the operator. And the repressor sticks out from the operator, because it is the thing that kind of determines all the stuff that happens. Oh my goodness. Repressor. Need to give myself more space here. Professor is here. Got the start. And then the actual coding region. And then stop.
Want to proceed in this direction and transcribe this entire area of the DNA, but the repressor doesn't allow it to do that. It just sits there and blocks transcription. And so again, this is the default situation that's in the cell. It uses glucose as an energy source, and transcription of these genes is blocked. Well, what happens when there's suddenly a lot of lactose in the cell? So let's just draw some lactose molecules. I'm going to draw them as these little triangles, although that does not adequately represent what lactose looks like, but for now we'll just draw it like that. So we have a bunch of lactose floating in the cell. So what's going to happen is that a lactose molecule will attach itself to the repressor protein. This changes the conformation of the repressor protein somewhat, and that causes the repressor to come off the operator site. So let's just get rid of our repressor. And let's put it, well, over here for now, together with the lactose that was attached to it. Now, the path of RNA polymerase is open, so it moves in this direction and transcribes all these genes. We make beta-galactosidase, we make lactose permase, and we have all the enzymes that we need to metabolize lactose. Well, what happens when the level of lactose goes down and we broke down more lactose? So let's get rid of some more lactoses. Well, now, while they're all taken care of, including this lactose that was attached to the repressor, and when the lactose comes off of the repressor, it changes the conformation of the repressor and causes it to go back onto the operator site. So let's put him back where he was. Now, you can see the RNA polymerase again is blocked, and so there is no more transcription happening right over here. So let's just connect this to what we spoke about in the beginning. We said that the regulation of gene expression happens at the level of transcription. We only transcribe those genes that we need. And this is exactly what's happening over here. In the absence of lactose, these genes are not expressed and we're saving the energy. But when we have lactose around, these genes are expressed and we have the enzymes necessary for the metabolism of lactose.
Hold up, I have made a slight mistake. Let's do this instead. Move this out of the way. Give myself more room to expand on what I've already done in terms of transcription. Like that. Just bring this all over here. Okay. Operator is in. Perfect. Nice. Alright, that basically covers what was just went over in that video. Okay. And that is an example of a very famous operon. Alright, nice. We got it. In a structure and function again. Oh my lord. Oh, we're getting into chromosomes. Here we go. Here we go. Oh, or we'll just dive deeper into the lac operon. Oops, that is not what I meant to do. That is not what I meant to do just now. There we go.
That the control of enzyme levels in cells occurs through the regulation of transcription. And they, along with another scientist, shared the 1965 Nobel Prize in Medicine for their work on what is called the lac operon. Now, a little bit of background first. An operon is a unit of genomic DNA containing a cluster of genes that are under control of a single regulatory signal, otherwise known as a promoter. And these genes are co-transcribed into a single mRNA strand and either translated together or undergo transplicing to create single mRNAs that are translated separately. So basically, genes in an operon are expressed either all together or not at all. Now, the operon that I've drawn here happens to represent the lac operon, and the lac operon is an example of an inducible set of genes which are responsible for importing and breaking down the sugar molecule lactose to use as a source of energy. So in the event that glucose, which is the ideal source of carbon and energy for a cell, if that's not available, then the cell has sort of a backup source of energy in the form of lactose. And you can see where the name lac operon comes from because it is named for the inducer molecule for the operon. And what do I mean by inducer molecule? Well, it is the presence of lactose that actually induces the transcription of the genes in this lac operon, which I'll explain in just a little bit. So there are three coordinately regulated genes contained in the lac operon. You have the lac-Z gene, which codes for an enzyme called beta-galactosidase, which is a cytoplasmic enzyme that cleaves lactose into glucose and galactose. The next gene is the lac-Y gene, which codes for lactose permease, which is a cytoplasmic membrane protein that transports lactose into the cell. And then finally you have the lac-A gene, which codes for digalactoside transacetylase. Now only the lac-Z and the lac-Y gene are actually needed for lactose metabolism. Lac-A is not as important in terms of understanding how the lac operon works. Now, besides these three structural genes, lac, Z, Y, and A, there are two regulatory sequences contained in the lac operon, and they are called the promoter, which promotes the transcription of structural genes, if you will, and then also the operator. And there are two other regulatory sequences that lie
Lac Z.
that are genes that encode for a repressor protein, and then you
I
have
too am lazy.
So these are the structural genes here, and then here are the regulatory genes.
Lac Z, lac Y, lac A. I don't even know what A does.
Meaning that it is transcribed at baseline, and that is just the default. And this regulatory protein binds to the lac operator.
Apparently it's useless. That's cool. It's all good.
RNA polymerase, which wants to bind here to the lac promoter. And this prevents and represses the transcription of these genes for lactose metabolism. Now, when glucose is not readily available to the cell, and an alternate source of energy is available in the form of lactose, then things start to change. First, lactose passively enters the cell at a pretty slow rate, and the metabolite of lactose, called allolactose, then binds to the repressor, and this alters the conformation of this repressor protein, or its shape, and it causes it to sort of loosen up and fall off the operator. Now, remember that the RNA polymerase is bound to the promoter immediately upstream of the genes. With the repressor now gone, RNA polymerase is free to sort of picture it rolling down to transcribe all of the three genes, leading to higher levels of the encoded proteins. So then you have lactose permease, which allows more lactose to enter the cell, and then you have more beta-galactosidase, which can break down the lactose into galactose and glucose to be used for the cell's basic metabolic needs. Now, what happens if both glucose and lactose are present? Which one does the cell prefer? Well, in that case, the transport of glucose actually blocks the transport of the inducer of the lac operon, the lactose, in a process that's called inducer exclusion. So actually, the transport of glucose into the cell leads to the formation of this protein intermediate that binds to the lactose permease and prevents it from bringing in any more lactose into the cell. So then you have decreased lactose, which leads to decreased repressor protein binding, so then the repressor protein then sort of latches back onto the operator there and prevents the transcription of the rest of the lac operon genes. Now, there are two key takeaway points from the lac operon model. The first is to realize that it is the interaction between the inducer and the repressor molecules that mediate gene expression. And the second idea is that the cell expends energy to make enzymes only when necessary. So there are inducible genes whose transcription is induced when a particular molecule is present. Is the interaction between the inducer and the repressor molecules that mediate gene expression. And the second process that's called inducer or the cell's basic metabolic needs. Now, what happens if both glucose and lactose are present? Which one does the cell prefer? Well, in that case, the transport of glucose actually blocks the transport of the inducer of the lac operon, the lactose, in a process that's called inducer exclusion. So actually, the transport of glucose into the cell leads to the formation of this protein intermediate that binds to the lactose permease and prevents it from bringing in any more lactose into the cell. Then you have decreased lactose, which leads to decreased repressor protein binding, so then the repressor protein then sort of latches back onto the operator there and prevents the transcription of the rest of the lac operon genes. Now, there are two key takeaway points from the lac operon model. The first is to realize that it is the interaction between the inducer and the repressor molecules that mediate gene expression. And the second idea is that the cell expends energy to make enzymes only when necessary. So there are inducible genes whose transcription is induced when a particular molecule is present.
Okay. I never want to look at that operon ever again.
So the regulation of gene expression
Oh.
virtually any step in the process.
All right.
The initiation of transcription all the way to post-translational modification of
Oh
proteins.
my,
And every
it's
step
getting quieter every video. And I literally can't bring down my music any lower. Come on, man.
And it's the ability to regulate all these different steps that helps the cell to have the versatility and the adaptability of an efficient ninja, so that it expends energy to express the appropriate proteins only when needed. Or you can think of the cell as a lazy couch potato that wants to expend the least amount of energy as possible. So starting at the beginning of gene expression, let's talk about gene regulation as it pertains to DNA and chromatin regulation. Let's talk about the structure of DNA. DNA is packed into chromosomes in the form of chromatin, also known as supercoiled DNA. And so chromatin is made up of DNA, histone proteins, and non-histone proteins. And there are repeating units in chromatin called nucleosomes, which are made up of 146 base pairs of double helical DNA that is wrapped around a core of 8 histones. And there are 4 different types of histones within this structure of 8 you should be aware of. And they're named H2A, H2B,
Not like this.
H3,
okay.
That's just the nomenclature they've been given.
This is going to have to be a completely separate area.
By an enzyme called histone acetyltransferase, which I'll just abbreviate as HAT.
Oh, I have chromosomes right here. Nice. Okay.
So the acetylation of histones is sort of kept in balance by another enzyme that removes these acetyl groups, which is called histone deacetylase, or HDAC-HDAC. The acetylation of histones leads to uncoiling of this chromatin structure, and this allows it to be accessed by transcriptional machinery for the expression of genes. On the flip side of this, histone deacetylation leads to a condensed or closed structure of the chromatin, and less transcription of those genes. When these modifications that regulate gene expression are inheritable, they are referred to as epigenetic regulation. So when it comes to gene expression in DNA, you can basically think of DNA as coming in 2 flavors: densely packed and transcriptionally inactive DNA, which is called heterochromatin, and then less dense transcriptionally active DNA, which is euchromatin. And I like to think of heterochromatin as being densely packed and hibernating, like heterochromatin and hibernating both begin with H, kind of like a bunch of densely packed bears that are closed off in their cave for the winter, whereas euchromatin is waiting there with open arms, welcoming the transcriptional machinery to transcribe away. Now often you will see that histone deacetylation is combined with another type of DNA regulatory mechanism, and that is DNA methylation, and this occurs in a process called gene silencing. And this is a more permanent method of sort of down-regulating the transcription of genes.
We already
DNA
have
methylation
that.
involves the addition of a methyl group, which is a carbon with 3 hydrogens,
Oh my God.
to the cytosine DNA nucleotides by an enzyme appropriately called methyltransferase. And this typically occurs in cytosine-rich sequences called CpG islands. Don't forget that cytosine pairs with G, guanine, so that's why there are CG islands that you'll find. DNA methylation stably alters the expression of genes, and so it occurs as cells divide and differentiate from embryonic stem cells into specific tissues. And so this is essential for normal development and is associated with other processes such as genomic imprinting and X chromosome inactivation, topics for another discussion. And abnormal DNA methylation has been implicated in carcinogenesis, or the development of cancer. So you can see how the normal functioning of DNA methylation is a critical regulatory mechanism for our cells. Now, DNA methylation may affect the transcription of genes in 2 ways. First, the methylation of DNA itself may physically impede the binding of transcriptional proteins to the gene. And second, and likely more important, methylated DNA may be bound by proteins known as methyl CpG binding domain proteins, or MBDs for short. Now, MBD proteins can then recruit additional proteins to the locus or a particular location in a chromosome, certain genes such as histone deacetylases and other chromatin remodeling proteins, and this modifies the histones, forming condensed inactive heterochromatin that is basically transcriptionally silent.
Seems legit.
So to the locus or a particular location in a chromosome.
Acetyltransferase. Alright. Transferase. Okay. Miro is not working with me today. There we go. Okay. Oh my god, come on. This almost looks workable. Nice. 10. Okay. Shoots off towards histone deacetylase. Alright, I think we got it.
So what makes a cell that's located inside of your nose responsible for smelling, say, a slice of pizza, look and act differently from a cell that lines your gut and is responsible for absorbing the nutrients from that pizza? They have the exact same DNA, so the differences can't be attributed to that fact alone. The answer actually lies in the expression of that DNA, which genes are actively transcribed and which ones aren't. And there are several ways in which gene regulation occurs at the level of
I
transcription.
should put
and so
like
we're going
a
to be talking about the ones
sticker.
here. Now,
It
let's
says
draw
subscribe
a
on
hypothetical
this.
gene here, and associated with this gene is a sequence upstream, so towards the 3' region of the antisense strand, also called the template strand. And this sequence is called the promoter. And there's another sequence in between the promoter and the gene called the operator. The operator is the sequence of DNA to which a transcription factor protein can bind, and the promoter is the sequence of DNA to which the RNA polymerase binds to start transcription. Now, first off, in prokaryotes, we have what are called general transcription factors, which
I'm
are
sorry,
class of
what
proteins
is
that
the
bind to
TF?
specific sites on DNA to activate transcription.
The transcription.
General
Okay.
transcription factors plus RNA polymerase and
Literally
another
just
protein complex
answered
called
it.
the mediator
Nice.
multiple protein complex constitute the basic transcriptional apparatus, which positions RNA polymerase right at the start of a protein coding sequence or gene, and then releases the polymerase to transcribe the messenger RNA from that DNA template. Now, there's another type of DNA binding protein called activators, and these enhance the interaction between RNA polymerase and a particular promoter, encouraging the expression of the gene.
Okay.
And activators can do this by increasing the attraction of RNA polymerase for the promoter
Doing
through
everything.
interactions with subunits of the RNA polymerase, or indirectly by changing the structure of the DNA. An example of an activator is the catabolite activator protein, or CAP, and this protein activates transcription of the lac operon in E. coli. In the case of the lac operon in E. coli, cyclic adenosine monophosphate, or CAMP, is produced during glucose starvation, and so this CAMP actually binds to the catabolite activator protein, or CAP, which causes a conformational change that allows the CAP protein to bind to a DNA site located adjacent to the promoter. And then this activator, the CAP, then makes a direct protein-to-protein interaction with RNA polymerase that recruits the RNA polymerase to the promoter. Now, enhancers are sites on the DNA that are bound to by activators in order to loop the DNA in a certain way that brings a specific promoter to the initiation complex. And as the name implies, this enhances transcription of the genes in a particular gene.
Oh, I
system.
freaking love
while
enhancers.
Enhancers are usually what are called
Oh,
cis-acting
shit,
cis means
okay.
we're acting on the same chromosome. An enhancer doesn't necessarily need to be particularly close to the gene that it acts on, and sometimes
It's
it's not
like-
located.
They'll flip over the DNA. Look at this dude having to flip over the DNA to ask a certain polymerase out for dinner or something, and it's just like, yo, what? All right, I respect it. Thinking outside of the box here, thinking outside of the box, and winning, and gaming. What was I typing here? Activators. All right. Although I feel like, aren't activators typically on the operator, or am I missing something here? Activator. RNA polymerase here. We have this here. Transcription factors. Place that somewhere here as well. So from my understanding, that's just stuff that we have already included on this map here. Like, this is just the broader name for activators and repressors, right? Oh god. I hope it is, or else I am very confused. Description factor. Alright, now we get to the legendary Legend 27 enhancer. Enhance. General transcription factors, no. Yeah, we're
on
good.
the same chromosome. Enhancers don't act on the promoter region itself, but are actually bound by activator proteins. And these activator proteins can interact with that mediator complex I mentioned earlier, which recruits RNA polymerase and the general transcription factors, which then can lead to transcription of the genes. So here I've drawn a little schematic of what it might look like to have the enhancer actually change the structure of the DNA so that the DNA is now looping around. Here you still have your promoter sequence, the operator sequence, the gene sequence, and the enhancer sequence, and having the DNA looped in such a way so that you then recruit RNA polymerase, the transcription factors, the mediator protein complex, and then you have transcription begin of this gene here. Now let's talk about repressors. Repressors are proteins that bind to the operator, impeding RNA polymerase's progress along the strand and thus impeding the expression of the gene. Now, if an inducer, which is a molecule that initiates gene expression, is present, then it can actually interact with the repressor protein in such a way that causes it to detach from the operator. And then this frees up RNA polymerase to then transcribe the gene.
Okay, wait a
for the
minute.
DNA strand.
I clearly do not understand what transcription factors are actually. In like a definitional sense. Ooh, okay. Hello. You know what? I should just have a separate like window just for Google. Because I can't just rely on everything that I'm seeing on this thing, because I need to search up stuff separately, especially if I get lost in something that we might have covered before, but then, you know, it's been a second, stuff like that. Proteins involved in the process of converting and transcribe, that doesn't help me. Transcription factors include a wide number of proteins, excluding RNA polymerase, that initiate and regulate the... Wait, what song is this? Okay. Cool, cool, cool. I was very curious about that because this soundtrack is from the same composer that does Megalobox, and this is a very similar track to one that's from Megalobox. A little bit better because it has more style, but the protein that controls the rate of transcription. I am literally just looking for... Okay. Yeah. Transcription factors work alone or with other proteins in a complex by promoting as an activator or blocking as repressor. Okay, I was right. I was correct. So it's literally everything that isn't the RNA polymerase that is being interacted with for the purposes of making genes more expressible
One example
or
of a
less.
repressor protein is the repressor protein associated again with the lac operon operator, which prevents the transcription of genes used in lactose metabolism unless lactose, which is the inducer molecule, is present as an alternative energy source. Now, silencers are regions of DNA that are bound by repressor proteins in order to silence gene expression, and this mechanism is very similar to that of the enhancer sequences that I just talked about. And similarly, silencers can be located several bases upstream or downstream from the actual promoter of the gene. And when a repressor protein binds to the silencer region of the DNA, RNA polymerase is prevented from binding to the promoter region. Now, a few notes about the differences between prokaryotes and eukaryotes when it comes to transcriptional regulation. In prokaryotes, the regulation of transcription is really needed for the cell to be able to quickly adapt to the ever-changing outer environment that it is sitting in. The presence, the quantity, the type of nutrients actually determines which genes are expressed, and in order to do that, genes must be regulated in some sort of fashion. So a combination of activators, repressors, and rarely enhancers, at least in the case of prokaryotes, determines whether a gene is transcribed. In eukaryotes, transcriptional regulation tends to involve a combination of interactions between several transcription factors, which allows for a more sophisticated response to multiple conditions in the environment. And another major difference between eukaryotes and prokaryotes is the fact that eukaryotes have a nuclear envelope, which prevents the simultaneous transcription and translation
Eukaryotes
of
are so
gene, and this adds an
sophisticated.
extra spatial and temporal control of gene expression.
Alright. This is the last video before I call it again today.
Let's talk about post-transcriptional regulation,
No,
which
no,
is
this last
regulation
one.
basically once DNA has been transcribed into mRNA. And I've drawn up this little schematic here, and it kind of just
Going
shows
back
you
to
how a
exons and
DNA strand
introns.
has a corresponding RNA strand and then the mRNA strand afterwards. And I'll sort of explain what all the different colors
We
and words
only
mean in
care
just a
about
moment.
the
So once DNA is
exons,
transcribed
dude.
by RNA polymerase into the corresponding RNA strand, this RNA strand needs to get what I call a tidy little haircut and then don some protective outerwear before it can leave the comfort of the nucleus for its big debut into the cytoplasm in the form of a fully processed messenger RNA or mRNA strand. Now, keep in
Along
mind
with
that
all
this
the
form of
stuff that
regulation
comes
occurs
with
in
that,
eukaryotes
yeah.
only, and this modification also helps to stabilize the mRNA to protect it from premature degradation before it gets translated into a protein. So, as you can see here, DNA gets transcribed one-to-one, base for base, into RNA. And you can see here that there are sections of the RNA that ultimately make it into the finished mRNA, these short segments which are termed exons. And they are the sequences that code for the ultimate protein product. And then there are short non-coding segments of RNA that get cut or sliced out. And this would be the haircut that I alluded to earlier. And so these are called introns. And this is
Video
accomplished
editing,
by
but
a large
for
molecular entity called the
genes.
spliceosome. So the spliceosome
All
binds on
coined the
either side of an intron,
term
moves the intron
gene
into a circle,
editing.
and then cleaves it off, and then ligates the two cut ends of the exposing exons together.
I left my jokes way too
And
much.
an easy way to remember which sequences are exons, which ones are introns, is
What
that
can I say?
exons
I'm a
exit
funny
the
guy.
nucleus, and introns stay in the nucleus. So exons kind of stands for exit.
Spliceosome.
Now, even
I
though the
need to put
mRNA has gotten its nice little
spliceosome
haircut,
here.
it's not quite ready to leave the nucleus just yet. It has to grab what is called a 5' cap and a 3' poly A tail. Now, what are those things I just
Spliceosome
mentioned?
over
So,
here.
a 5' cap refers to changes at the 5' end of the mRNA. And remember that this is the phosphate end of the nucleotide bases in the mRNA. And some people like to remember this as F for 5' and for phosphate. So that's how you can kind of keep the two ends straight. And capping at the 5' end converts this end of the mRNA to a 3' end by a 5' to 5' linkage, which basically just protects the mRNA from exonucleases, which degrade foreign RNA. The cap also promotes ribosomal binding for translation and also helps the regulation of nuclear export of the mRNA. Now, the poly A tail, that goes on the other end, the 3' end of the mRNA, which has
Sounds
the
pretty
terminal hydroxyl
important.
group. And so what do I mean when I say poly A tail? Well, poly A tail refers to polyadenylation, in which multiple adenosine monophosphates, or basically adenine bases, are added to act as a buffer for exonucleases in order to increase the half-life of mRNA and, again, protect it from degradation. And so the purpose of the poly A tail is really very similar to the 5' cap.
Okay, just searching up spliceosome just to make sure that I got this correct. Spliceosome. Large RNA protein complex that catalyzes the removal of introns from rearm mRNA.
Which is basically to protect from degradation, help with promoting translation, and regulating nuclear export. The poly A tail also does one more thing. It kind of just helps with transcription termination for the RNA polymerase that's transcribing the messenger RNA. Polyadenylation is catalyzed by an enzyme called polyadenylate polymerase, which adds the adenosine monophosphates using adenosine triphosphate as a substrate. And the poly A tail is built until it's about 250 or so nucleotides long. So overall, the 5' cap and poly A tail help to stabilize the mRNA for translation. That's the key point to take home from here.
Yep.
So once the mRNA has donned its cap and tail and had its intron spliced out, it's now ready to exit the nucleus to be translated into a protein. Now,
As we
additionally,
talked
there's
about
one more type of
earlier,
RNA regulation called RNA editing,
trying
which
to
is a process
avoid
that results in sequence variation of RNA
the
molecule and is catalyzed
enzymes
by various enzymes.
around
RNA
the
editing is relatively
eukaryotic
rare, and these events may include insertion, deletion, and base substitution of nucleotides within the edited RNA molecule. Now, one of
cytoplasm
these enzymes is
from
called adenosine
freaking
deaminase
out and
acting on RNA,
eating
or
all the
ADAR enzymes,
RNAs.
which convert specific adenosine residues to
That would be
inosine
bad.
in an mRNA molecule by hydrolytic
That'd be really
deamination.
sad, actually.
Another type of editing is called cytidine deaminase acting on RNA, or CDAR, which involves deamination of cytosine to uridine by cytidine deaminase.
Sorry, do I
RNA
need to know
editing is
this?
currently being extensively studied in relation to infectious diseases because the editing process alters viral enzymes and their function. So, kind of an exciting new emerging concept in
Oh,
post-transcriptional
this is just
regulation.
switching the stuff from
Emerging concept in post-transcriptional regulation.
DNA to... Wait, no. Right. Okay, I'm gonna have to listen to that explanation again.
Regulation called RNA editing, which is a process that results in sequence variation in RNA molecule and is catalyzed by various enzymes. RNA editing is relatively rare, and these events may include insertion, deletion, and base substitution of nucleotides within the edited RNA molecule. Now, one of these enzymes is called adenosine deaminase acting on RNA, or ADAR enzymes, which convert specific adenosine residues to inosine in an mRNA molecule by hydrolytic deamination. Another type of editing is called cytosine
What in the
deaminase
world
acting
is
on RNA, or
inosine?
CD.
That sounds badass, but like, what is it? I have never heard of this thing in my life. The nucleoside that forms when carboxytine is attached to a ribose ring. Oh, it's found in tRNAs. Oh, okay. Okay, I see. Hearing gene translation modulation of widespread post-transcriptional modification and transfer RNAs and messenger RNAs. Hold up.
CDAR, which involves deamination of cytosine to uridine by cytidine deaminase.
Hey, does what? Okay, I understand this. That's what RNA typically has, which is uridine. Adenosine is a modified adenosine. I have literally never met this nucleoside in my life. Okay. Cool. Things have changed, and now I am reconsidering my entire life choices. Okay. ADAR. Alright. ADR and CDR. DAR. CDAR. So do I just type out adenosine or A to I? There we go. That's not going to be confusing. Oh god. I can imagine myself coming back to this just like a month later, being so confused. Oh, don't do it, Nick. Don't be confused. It's alright. See, this makes sense. C to U? That makes sense. A to I? I've never met this man in my life. Okay. Boom. Alright. We got it. We got there. Somehow.
Diseases because the editing process alters viral enzymes and their function.
Alright.
So, kind of an exciting new emerging concept in post-transcriptional regulation.
Emerging concept. Alright, so maybe it's something that I haven't been expected to learn yet, but I'm going to just for this. Why not? Alright. For now, though, I'll see y'all later. Bye-bye.